Hi, and welcome to the VieSID Press-release. In this video, we report on the dental literature published in February 2022. We've chosen articles from a selected list of journals that we think might be of interest to the VieSID community. So let's begin. In the Annals of Anatomy, Medze et al. Described the third layer of the Masseter muscle that was previously inconsistently reported in the literature. They based the description on cadaver dissection, CT and MRI image data, and histological data. They could reliably demonstrate the presence of a coronoid part of the masseter running from the medial surface of the zygomatic process of the temporal bone to the root and the posterior margin of the coronoid process. The coronoid part of the masseter is innervated by branches of the masseteric nerve. Based on its diagonally running fibers, they suggested that the coronoid part of the masseter stabilizes the mandible by elevating and retracting the coronoid process. In the same journal, Kuchukuen et al. Demonstrated the TMJ innervation by dissection of 20 cadavers. After exposing the nerves, they noticed that the auriculotemporal nerve, ATN in their figures, is the main nerve innervating the TMJ, with a number of branches varying from 1 to 3 and most commonly 2. The ATN reaches the TMJ capsule and retrodiscal tissue posteromedially and posterolaterally, turning around the condyle neck. TMJ branching of the mandibular nerve was observed in 13 of the 20 individuals and innervated the TMJ medially. The masseteric nerve innervates the TMJ capsule anteriorly, usually with one branch. The innervation of the anteromedial surface of the TMJ capsule by the posterior deep temporal nerve was found only in 3 out of 20 individuals. The authors went on discussing the possible clinical implications of the TMJ innervation. For example, in TMJ hypermobility, anterior dislocation of the TMJ, or interventions affecting the anterior region of the joint, the masseteric nerve may be injured. The TMJ branch of the mandibular nerve, as well as the auriculotemporal nerve, might suffer compression injuries in case of medial dislocation. In the Japanese Dental Science Review, Cruz et al. Reviewed articles investigating the genetic profile of TMD and primary headache disorder to find out whether there could be genetic commonalities in the individuals suffering from these conditions. Through this review, Cruz et al. Found that 3 different genes were assessed with regard to both TMD and primary headache and went on discussing them. The first, the MTHFR gene, encodes an enzyme involved in the homocysteine metabolism necessary for processing vitamins of the B complex. This gene has been highly investigated as a generic risk factor for migraine. And it is also known that nutritional deficiency of vitamin B can induce myofascial dysfunction and pain. However, Cruz and colleagues did not find enough evidence to support the idea that this gene is common to both conditions. The COMT gene encodes an enzyme present in the metabolic degradation of numerous neurotransmitters and is associated to chronic pain conditions. COMT genes polymorphisms were shown to play a role in the pathogenesis of TMD. COMT gene was found also to be associated to lower widespread pressure pain thresholds. Nonetheless, the authors concluded that this gene should not be considered as common genetic background to TMD and primary headache. For the estrogen receptor 1, ESR1 gene, results were more substantial and associations to both TMD and primary headache were found. Severity of pain seems higher in females for many body regions, including face and TMJ. Moreover, the menstrual cycle seems to worsen TMD pain. The peak of pain is experienced during the phase of rapid estrogen fluctuations. And similarly, there is evidence that a late luteal decrease in estrogen is both a trigger and an aggravating factor in the migraine pain intensity. So clinically, association can be explained by the presence of estrogen receptors alpha in the areas of the brain, as well as in the trigeminal ganglion, which are more expressed in females than in males. Last February, the Journal of Oral Rehabilitation has published a special issue on orofacial pain in China, including both reviews and original articles by clinicians and researchers from various Chinese institutions. Here we report about some of these contributions, but however, we applaud the Journal of Oral Rehabilitation and all its contributors for this interesting issue. Becca Liu et al. Investigated the neuroplasticity of the cortical motor control of the masseter muscle in relation to orthodontic treatment. They recruited 15 Engel class 2 patients who received orthodontic treatment and used then transcranial magnetic stimulation to measure the amplitude of the motor evoked potentials, which reflect neuronal excitability in the motor cortex. To elicit a muscle response, patients were asked to bite on a special bite device placed between the front teeth to activate the muscle. Evaluations were made at baseline, before ligature of the arch wire, 1 day after ligature, and then again at 7 and 30 days. The orthodontic treatment caused an increased excitability of motor cortex of the masseter muscle. But as the orthodontic forces gradually decreased, the response of the periodontal mechanoceptors, the motor threshold of the masseter muscles, and the excitability of the cerebral cortex neurons returned progressively to normal. The same results are illustrated here through color maps. These demonstrated the neuroplasticity of the cerebral cortex, which adapts to nociceptive stimuli. My colleagues investigated the condylar positions in patients with and without preferred chewing side. They gathered the measurements from CBCT images using a geometric construction with equiangular space radii as in this illustration. They find out that in patients with a preferred chewing side, either a IPSI or contralateral condyle was more posteriorly located than in patients with bilateral mastication. However, the more posterior position of the condyle was not associated to increased pain. The authors concluded that condylar position is not a predictor to unilateral chewing side related to TMD from that not related to a TMD. Although this study deals with an important topic, I find that some biases could have influenced both the results and the author's conclusions. In fact, there were no subjects with unilateral chewing in this study that weren't affected by TMD. The patients were selected based on the presence of unilateral TMD symptoms rather than on preferred chewing side. Moreover, 2D linear measurements from a CBCT slice were used, thus losing the benefit of working with 3D images. Anyway, I see interesting results already emerging from patient selections of this study. In fact, of the 90 patients with unilateral TMD symptoms that they recruited, 77% chewed on a preferred side. That is 3 out of 4 patients. This is striking. The study group, namely the TMD group, also had a more posteriorly positioned condyle, which should also raise a flag. In a review paper, Wren and colleagues screened 27 articles testing the effectiveness of low-level laser therapy. The results of the meta-analysis revealed that low-level laser therapy with different wavelengths is effective in reducing TMD pain. Higher efficacy resulted from higher wavelengths ranging from 910 to 1,100 nanometers. An effect of low-level laser therapy was revealed in the short term, just after treatment and after a month from treatment, but the reviewed articles did not provide evidence for therapeutic effects in the long term. Shasha Lui et al. Investigated the effect of platelet-rich plasma therapy and platelet-rich plasma therapy combined with physiotherapy in patients with TMJ osteoarthritis. Platelet-rich plasma has a 3 to 5 times higher platelet concentration than normal human plasma. It contains several growth factors and bioactive substances and is one of the core rehabilitation and regeneration therapies for osteoarthritis in large joints. The authors consider 40 patients diagnosed with osteoarthritis based on the DC-TMD and image diagnostic, including both CBCT and MRI. 19 patients received platelet-rich plasma therapy and 21 received the combined plasma and individualized comprehensive physical therapy 2-3 times per week for 3 weeks after the injection. The pain intensity, maximum mouth opening, and TMJ sounds were evaluated before treatment and at 1, 3, and 6 months after injection. Pain intensity decreased significantly and mouth opening improved significantly in both groups. Also, some improvements were noted in the severity of TMJ sounds. Although improvements were found in both groups, the combination of platelet-rich plasma and physiotherapy was the most effective. In a further article, Sao described different experimental settings in which it has been demonstrated that artificial alteration of occlusion induced orofacial pain. Sao described the mechanism of orofacial pain related to occlusal disharmony based on animal studies. He explained also the pathological changes extending from the peripheral tissues to the central nervous system occurring in case of muscular pain directly induced or induced through the insertion of an occlusal interference. In this image, borrowed from Cecil 2011, you can follow the orofacial pain path explained by Sao. Additionally, the author concluded that myofascial pain related to occlusal disharmonies is also closely related to dental treatment and can seriously affect a patient's quality of life. Thus, it deserves attention of the dental community. We selected a further study by Mo and colleagues exploring the involvement of the astrocytes in the rostral ventromedial medulla in case of orofacial hyperalgesia induced experimental by artificial occlusal interferences and after removal of the occlusal interferences. So let's first clarify some anatomical terms. The astrocytes are starry neural cells with a number of processes or ramifications, each of which is in contact with the neuron synapses receptor complex. These cells are important in maintaining regulatory functions of the brain, nourish and support the neurons, permitting also catabolic activities of the neurotransmitters. A curiosity, the analysis of Einstein's brain revealed that it takes an increased number of astrocytes to make a genius. The rostral ventromedial medulla is represented by a group of neurons located in the brainstem, indicated in red in the animation. It is involved in the relay of sensory information between the spinal cord and brain, including descending pain modulation. Now back to the experiment of Mo and colleagues in which they established a rat model of sustained orofacial hyperalgesia. They placed the metal crown unilaterally to generate a persistent dental occlusal interference. Then induced pain by exerting a pressure on both ipsi and contralateral masseters and assessed muscle pain by measuring the tolerance to pain before head withdrawal. In some rats, the interferences were removed at day 3 and reaction to pain was studied at day 6. In others, rats, the interference was kept till day 8 and pain was studied at day 14. And now let's see the first outcomes. The bars in the histogram reflect pain tolerance. So on the very left, the bars for the control rats, so with pseudo-interferences around the dental crown, but not on the occlusal surface. As you can see, the experimental interference generated a significant decrease of tolerance to Masseter stimulation in both study groups, in the center and right side of the histogram. But what is the role of astrocytes in the rostral ventromedial medulla in the modulation of pain? My colleagues found significantly different activation in response to induced pain only at day 8. Thus, they performed a further experiment and observed an increase in a tolerance to pain in these rats if they used the blocker of the astrocyte signaling, thus demonstrated involvement of astrocytes in the rostral ventromedial medulla in maintaining of orofacial hyperalgesia after prolonged induced pain. Liu et al. Explored the effects of Genestein in reducing muscle pain. So now let's put this statement into context. It is known that females are affected by TMD in a 3-to-1 ratio with respect to males. In fact, estrogen has been proven to play a role in the pathophysiology of pain. Now, Genestein is an isoflavonoid contained in different types of legumes. It is a phytoestrogen known for its analgesic properties in case of inflammatory pain, neuropathic pain, and migraine. Thus, Liu and colleagues wanted to test whether Genestein could be considered for treatment of orofacial pain. Liu and colleagues performed the experiments on rats, to which they induced pain of the masseter muscle by inserting an occlusal interference, a metal crown. They measured the level of pain by observing the head withdrawal threshold when the masseter was mechanically stimulated, indicating the level of tolerance to pain of the rats. Evaluation was performed before the insertion of an experimental occlusal interference and afterwards for 28 days using both male and female rats to explore possible sexual dimorphism in the response to pain. The study group showed a decreased tolerance to masseter stimulation with females in both study and control groups less tolerant to pain than males. Moreover, we can observe that the induced hyperalgesia was higher in the first week after insertion of the artificial interference and improved over the second week, stabilizing in the third and fourth weeks. The response to masseter stimulation in female rats varied depending on the phase of the estrous cycle, with lower tolerance to pain during proestrus. In this histogram, comparable for the analysis of the right and left masseters, results for ovarectomized female rats are shown. The rats receiving Genestein in groups D to F showed increased tolerance to mechanical stimulation of the masseter. Liu et al. Demonstrated also the co-occurrence of estrogen and its receptors in the trigeminal ganglion. Additionally, they showed that the activity of the estrogen receptors in the trigeminal ganglion was downregulated by Genestein. In summary, the present study demonstrated sex difference in masseter mechanical hyperalgesia in rats induced by experimental occlusal interference. Female rats displayed an enhanced sensitivity to occlusal interference. Estradiol replacement exacerbated masseter hyperalgesia in ovarectomized rats, while Genestein could antagonize the effect of estradiol in inducing hyperalgesia. Lee and colleagues have investigated the chemokine signaling pathways in relation to the pathogenesis of neuropathic pain. Chemokines are neuromodulators, mediators of the neuron glial cell crosstalk, depicted here in an illustration by Del Rio et al. 2015. Chemokines are considered to play a role in some neuropathic pain conditions and neuroinflammation processes. In particular, this study focused on the chemokine membrane receptor called CXCR3 and its ligands, CXCL9 10 and 11, which were previously detected in the spinal dorsal horn after nerve ligation in chronic constriction injury. Here you see the outcomes of the study for 4 groups of mice, the wild type and the knockout mice, the latter being the mice in which the gene encoding for CXCR3 chemokine membrane receptor was inactivated. These were further divided into 2 groups, the study group in which ligation of the sciatic nerve was performed, and the sham in which the sciatic nerve was exposed but not ligated. Liu et al. Demonstrated that expression of CXCR3 and its ligand CXCL11 was upregulated in the spinal cord after chronic constriction injury. Moreover, they show that CXCR3 signaling was involved in pain modulation, since in knockout mice with chronic constriction injury, pain response was inhibited. Therefore, they suggested that CXCR3 could be a potential therapeutic target for intractable chronic pain. This presentation ends here. I invite you to contact us in case you want to know more about our activities. Otherwise, be ready for next press release. And best wishes to all.